Welcome to the lesson, summary lesson on the power of the cholera toxin material. This lesson is really all about exotoxin-based diseases, so they're all going to be bacterial. There are exotoxins that are found in certain fungi and other organisms, but they're primarily bacterial, and that's what we are going to focus on. So the overall summary kind of setup disease here is cholera. There are several videos that I think do a really good job of talking about the spread of cholera, the prevention of cholera, its impact in a population and around the world. So I'm not going to go into detail of that. Make sure to watch those videos. But it's primarily spread through contaminated water. You ingest the bacteria. The bacteria that causes cholera is Vibrio cholerae. That's the epidemiological agent that and I do want to address some misinformation, that there's some confusion that exists about Vibrio. Vibrio is a genus. There are a bunch of different species of Vibrio. They really like warm salt water, so you can go out to places like the Gulf of Mexico and find Vibrio. Only the species cholerae is what causes cholera. So Vibrio cholerae is the cause of cholera. The other species of Vibrio can cause various things, can cause GI issues, can cause wound diseases, and can be found, like I said, in the Gulf of Mexico and other places that we're all familiar with. But, if you're reading a newspaper article about an infection from, Galveston or something, they talk about a Vibrio. Unless it's Vibrio cholerae, then we're not talking about cholera. We're talking about a different disease presentation. Okay? Okay, so Vibrio cholerae has several really neat virulence factors. We're going to focus on four. The first two, flagella and fimbriae, are all about swimming to your mucosal lining of your gut and then attaching there. I do want to warn you that for many of the resources that talk about Vibrio cholerae, it'll use the term pili instead of fimbriae, but these are truly fimbriae in the attachment sense. And then once they are attached, they stop producing the flagella and fimbriae proteins, and they start shifting their energy into production of what's called a biofilm. Living film, right? A living film. Biofilms are found in various infections and various natural places in your body. The surface of your teeth is a classic example of a biofilm, where organisms stick onto a surface and then they produce a thick layer on top of them like a blanket of various sugars and proteins. Think of like a slime layer. It's thick, sticky layer on top of them, and it serves to protect them from the immune attack, protects them from chemicals in the environment, protects them from antibiotics in the environment. And one neat thing with Vibrio cholerae is when the bacteria are in this biofilm, they actually start releasing chemicals and communicating with each other and start cooperating almost as a group instead of individual bacteria, unicellular organisms. So once they've established this protective biofilm, they start really, multiplying themselves, and you start seeing large amounts of the cholera exotoxin being produced. And the effect of the cholera exotoxin is described in several of the different videos. There's one video I think that this is a screenshot of that does a really good job of actually showing at a molecular level what the exotoxin does. The part that you need to get, or the part that I want to emphasize, is that this comes back to tonicity. Remember, we talked about osmosis and hypertonic, hypotonic, isotonic, that stuff. What the cholera toxin does is it causes dehydration by messing with the salt levels of the cell. So it's not that it's sucking the water out directly, it's that it's causing a shift in osmosis, which causes massive water loss and death because of dehydration. So you can watch those videos and get those details. The next part of this lesson moves into the history of cholera and some epidemiology. And this disease in particular has a very, very, very rich history filled with several really important scientists, several firsts, several kind of big, high impact historical things. So There's a great video that I have linked for you, all about John Snow and the big London outbreak of cholera, and he was really the first to be a disease detective, if you will. So epidemiology is the study of epidemics. It's the numbers, the locations, the causes of infections. And it's really kind of the heart of public health. And so he really established this entire class of science and class of public health with the cholera, with the big cholera outbreak in London. Several other very high impact, important researchers were involved with cholera. The two that are kind of credited for discovering the actual etiological agent, the actual Vibrio cholerae, are Filippo Piccini and Robert Koch. Filippo Piccini was an Italian scientist who discovered this bacteria, but he wasn't quite as famous as a microbiologist as Robert Koch. So even though Robert Koch discovered it, isolated it many years later, he kind of got most of the attention. So he's still given credit for it. He actually went on to do a significant amount of work on the spread of the bacteria, on treatment of the bacteria. Robert Koch won a Nobel Prize in 1905 because he had worked so hard in what we call the germ theory of disease. He was really one of the first people to show that a specific organism causes a specific disease. And he was able to isolate organisms from sick people and show that if you give that organism to somebody who's not sick, they get sick. And so he was able to do that with over 20 different diseases where he showed the actual organism. And really neat thing was he was one of the first people to really figure out how to culture bacteria. He created media to let the bacteria grow, so he could literally sample an organism from a person and grow up what he found in that person and show that the isolated organisms from that person caused disease. And that whole process of isolating the cause, showing that cause is that organism or that mutation or whatever it is actually replicates that disease in an unaffected organism. It's called Koch's postulates, and that's still what is shown today. If you mutate this, it causes this disease, or if you give a person this infection, it causes this disease. And he really kind of championed that and created that whole thought process. So very important man, beyond just cholera. A couple of other very important scientists who were kind of in the same time frame, working in similar places or on similar things. Joseph Lister is a really important man for public health and microbiology. He was actually a surgeon. He was not a microbiologist, but he was also in London, and he was hearing this information from John Snow, from Robert Koch about these invisible organisms that can cause disease. So he really fundamentally created the concept of aseptic technique, the concept of cleaning tools before surgery, of changing your clothes, of washing your hands, of having a clean environment for surgery. Before him, people were dying more of infections than they were of the actual diseases that they were going in surgery for. Surgery was almost a death wish because of infection. So he really established the thought process in the surgical world of, take these organisms, the concept of these organisms, and try to reduce them. Remember, this is long before antibiotics. Another and most famous microbiologist is Louis Pasteur. So Louis Pasteur, Frenchman, so we've got some work going on in Italy and Germany and England and France, all about the same time. They're all working on similar concepts or similar diseases, literally. But what comes out of Louis Pasteur's work, his early microbiology work showed that there were invisible things that you couldn't see that were influencing your health, essentially. And then the invisible things, he started working on actually finding those invisible things. And then by the end of his career, he actually ended up developing several vaccines. And he was really kind of the founder of the inactivated vaccine. Remember, those are the dead organisms. So he created a chicken cholera vaccine, which unfortunately for him, chicken cholera is not the same etiological agent as human cholera. It's a bacteria they actually named after him called Pasteurella that is mostly an animal infection. It can cause human infections, but usually we find it in humans as bite site, kind of localized infections. He ended up creating a vaccine against anthrax and human rabies vaccines, among others. So, all these scientists about the same time over in Europe, all working to unlock the function of these microorganisms in disease. So really, really important time. Cholera is kind of at the heart of this amazing renaissance in microbiology and public health. Okay, so the rest of the lesson then moves into giving you examples of other exotoxin diseases. So there's a fairly detailed video on shigellosis, another bacterial disease, causes dehydration, not as dramatically as cholera though. The fatality is not nearly as high as cholera because it's a much slower, more progressive dehydration. Shigellosis does differ for cholera in a different way also. With shigellosis, one of the big signs of shigellosis is dysentery. So dysentery is bloody stool. So cholera is, almost clear stool. There's so much water coming out. With dysentery, you're looking for streaks of blood and mucus in the stool, and that's a really good sign that you might be looking at shigellosis. Again, spread through dirty water. But not nearly as rapidly fatal as cholera. And so, easier to treat as long as you can get that person antibiotics and get them clean water. You don't see it much in the developing world. The dysentery forms of shigellosis, which are caused by Shigella dysenteriae. We do have other species of Shigella here, but they usually just cause kind of a more subtle enterocolitis, upset stomach kind of diarrhea situation than this extreme dysentery. That's mostly in the areas of poor sanitation. Okay, another kind of GI example that is exotoxin-based is campylobacteriosis. The etiological agent is Campylobacter jejuni. This is very common in the United States. It causes a self-limiting diarrhea. That means that your body will be able to kick it eventually. You don't have to take antibiotics or, go get treated for it, although it usually takes a few weeks to really kind of get it out of your system. Frequent, diarrhea, maybe a little nausea, sour stomach, maybe a teeny bit of fever right at the beginning of it, but usually just pretty much sour, gross stomach for a while. You can differentiate campylobacteriosis compared to shigellosis because with campylobacteriosis, you're not going to see the blood. You don't get the dysentery effect. Plus, if you actually look at the organism, Shigella is a very plain gram-negative bacilli, just a really plain rod. Campylobacter jejuni is a gram-negative rod, but it's not just a flat little rod. It's actually an S-shaped rod. It's got kind of, I always call it the Superman bacteria because it's got this little S going on. It's pretty distinct under the microscope. So you take that fecal sample from the person and see these S-shaped guys, and you're like, yeah. So it's very commonly spread person to person. It also can be associated with contaminated animal products, especially cows and cattle, especially unpasteurized dairy products, unpasteurized milk and pasteurized cheese. It's pretty common to find Campylobacter in those. Okay, the next set of exotoxin diseases that we shift over to are various food poisonings. So I want to take a moment to really, really differentiate between a foodborne intoxication versus a foodborne infection. So an intoxication means that the organism is on the food producing toxin, and then you eat the toxin and your body responds to the toxin. Infection would be the organism is on the food, you eat the organism, it gets in your body, it establishes infection, it multiplies, it produces toxin. So the big difference here clinically, and kind of the obvious hallmark here, is timing. Because the exotoxin is already produced on the food with an intoxication, you respond really quickly, usually within a day, sometimes within a few hours. You eat that meal and a few hours later you're throwing up. As opposed to an infection where the bacteria has to get into your system and multiply, and you're talking a few days before you start really seeing your symptoms. So what we usually call food poisoning is typically an intoxication. I'm going to give you 2 examples of food poisoning in this lesson, and then we'll see another example of food poisoning in a subsequent lesson. So the 2 that we're going to talk about today are pretty mild. They are actually endospore-based foodborne intoxications. So we've got 2 different examples: food poisoning caused by Clostridium perfringens and food poisoning caused by Bacillus cereus. Both of these, Clostridium and Bacillus, remember, are 2 of the genuses that can produce endospores. So you've got an endospore that is in the ground usually, remember. And what is in ground? Well, things like grains, things like fruits and vegetables, and even also grass, where you have like a cow, for example, that eats the grass and can ingest in the endospore. So there are various ways that endospores can end up in our food supply. Remember, with endospores, they have an amazing ability to survive really extreme conditions like being frozen or being boiled, being cooked generally. You have to do some pretty extreme temperatures to harm an endospore. So the typical cooking method is not enough to kill an endospore. So if you have endospores in that, rice, for example, and you don't wash them well enough, the cooking process is probably not going to kill it. So the endospores will be in your finished product. So the way to limit foodborne intoxications, no matter what the source, is to limit the conditions that the food is stored in. So you need to limit the time. Don't give the organisms time to produce the toxin. So cook the food, eat the food. Or you need to limit the temperature. These organisms all mostly like warm temperatures. So, you cook the food and then you let it sit there and just be warm. And that's warm enough for the organisms to be happy and grow and continue. Another thing that you have to pay attention to is the oxygen. So endospores are actually anaerobes. They don't want oxygen. So you cook the food and then you seal it with a Tupperware lid, or with a dome on your buffet. And you limit the amount of oxygen that's getting in there. And that creates an anaerobic environment to allow these endospores to grow. So if you limit those things, doesn't have the time or the temperature of the oxygen, then the organisms won't be happy and they won't produce their endospores. Okay, so how do you differentiate between Clostridium perfringens food poisoning and Bacillus cereus food poisoning? Well, Bacillus cereus is a little more intense usually. Clostridium perfringens is so mild that you may not even really realize you have food poisoning. It's just one of those where like that food didn't sit very well. You ate and then the next morning you wake up and your stomach's feeling kind of gross, just sour and kind of diarrhea, kind of ugh. Bacillus cereus is usually more intense. Usually pretty intense vomiting or pretty intense diarrhea or both. And it usually happens within, a few hours. So you ate something weird at dinner and then you woke up in the middle of the night throwing up. Bacillus, just so you know, Bacillus cereus in the public health world has a nickname as fried rice food poisoning because Bacillus cereus is really strongly associated with rice. And buffets, like Chinese food buffets where you've got big containers of rice, can create that perfect storm of conditions where you've got them sitting under a heat lamp. It's sitting there for a while, giving it time. And then a lot of the times there'll be a sealed lid over it. Or even if there's not a sealed lid over it, if you got a giant bowl, right in the heart of that bowl, there's not much oxygen getting in there. So you can actually create an anaerobic environment that way. So I always tell people you don't have to be afraid of buffets, but go at a busy time. You want the food to be, cooked quickly and eaten quickly. And really good buffets, you'll notice that they come by and they stir. They oxygenate, and that can help with food poisoning also. Okay, so getting away from GI into some other areas of your body, we talked about scarlet fever already. Remember that strep throat. You've got the Streptococcus pyogenes bacteria on your throat. They can produce, certain strains can produce exotoxin. That exotoxin then sets off a set of symptoms called scarlet fever. So it's a sequelae of strep throat. So go back and review that in the immune balance lesson if you don't remember that. Another example of kind of a throat respiratory type exotoxin-based disease is diphtheria. Diphtheria is not a disease that we see much in the United States anymore because we have a very effective vaccine that's nearly eliminated it in the developed world. The etiological agent for diphtheria is a bacteria called Corynebacterium diphtheriae. And although the initial symptoms look just like strep throat, the bacteria are growing on the throat, causing a very, very similar set of symptoms to strep throat. If you actually culture the bacteria at that point, instead of assuming it's strep throat, if you actually look at it under the microscope, instead of seeing, streptococcus or gram-positive cocci chains, Streptococcus pyogenes, strep throat. Diphtheria, they're gram-positive rods that are really irregular length. Some are long, some are short. They're kind of interesting looking rods. So they don't look anything like chains of cocci. If the disease continues, if it's not treated with antibiotics and it continues, a few days in, you'll get the development of what's called a pseudomembrane, which is basically a film that forms on the back of the throat, and that is the effect of the exotoxin. And that is the real dangerous part of this infection. The strep throat type symptoms are not really that dangerous, but if allowed to continue, that exotoxin will take basically all the blood and the pus, all of the fluids of infection and start congealing it into this thick leather-like layer that fills the entire back of your throat, fills your esophagus. So it actually can be fatal, especially in children who just physiologically, they don't have much space there through their trachea, through their esophagus, and it can asphyxiate them. But also, it's very, very difficult to swallow. The peristalsis, the squeezing of those muscles, is really difficult when you've got that thick leathery film in the back of your throat. So you actually can die of starvation, essentially, because of difficulty swallowing. So we don't see this much in the United States because we have a really good vaccine against the diphtheria toxin. So even if the bacteria is here, if you can prevent the exotoxin from doing its work, then you just have strep throat-like symptoms, and it's no big deal. It's not so dangerous. Okay, another exotoxin respiratory disease is whooping cough, and there is a nice, pretty detailed video on whooping cough. This is one that I've chosen to expose you to a little bit higher level of clinical detail because we have been in a whooping cough outbreak. We've been in really an epidemic for about 10 years now, and we're still in it. So I want you to watch that video and really familiarize yourself with the clinical presentations of pertussis. The formal name is pertussis. That's the better name to know. Etiological agent, Bordetella pertussis. A very, very, very exotoxin-based disease. In fact, many strains of Bordetella pertussis produce 5 or 6 different exotoxins all at the same time. You don't need to know the detail of all the different exotoxins, but just to give you an example of how crazy these are, there's one that destroys all the cilia, just like the flu does, where it takes all the little paddles off of your respiratory tract so it can't flick out things. There's another exotoxin that causes mucus to be produced at much higher levels because the bacteria have evolved to actually to hide and be protected by the mucus. So your body is producing higher levels of mucus, plus it doesn't have the paddles to flick it out. So what do? You cough, 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 because your lungs start getting filled with mucus. And sustaining your oxygen levels gets harder and harder and harder. So we've actually had, babies especially can actually die from pertussis. Just not being able to maintain enough oxygen. So you can watch that video and get a little more detail on that. Okay, and the final exotoxin diseases that I want to present to you in this lesson are neurological ones. So we've got botulism and tetanus. There's a detailed video on tetanus, so I'm not going to go through it in great detail, but I do want to kind of compare botulism and tetanus together because they really are kind of siblings to each other. They're both caused by bacteria in the genus Clostridium, so they're both endospore-related diseases. Etiological agent for tetanus is Clostridium tetani. Etiological agent for botulism is Clostridium botulinum. Both of these, the endospore is the primary infectious agent. You get, with tetanus, you get a cut. The endospore gets deposited in your body. The bacteria start growing inside of your body, producing toxin. You start showing the signs of tetanus. With botulism, that can happen, but it's more commonly associated with food. Clostridium botulinum is found a lot on fruit and other sweet things like honey, around pollinated things. The bees will actually pick up the bacteria and bring it into honey, and then the honey is a wonderful environment. It's an anaerobic environment full of lots of food, so the bacteria flourish in honey. So you eat it, and then that can be the source of the botulism in your body. Both of these are exotoxin. The endospore comes in, starts producing the infection, exotoxin gets produced. The exotoxin actually attacks the exact same spot in our body for both of these diseases, even though they have opposite effects. So the exotoxin attacks right at our nerve muscle junctions where the neurotransmitters are released, the chemical signals that tell your brain, that allow your brain to tell your muscles what to do. So the botulism toxin, a nickname of which is Botox, but the botulism toxin goes in there and blocks acetylcholine signaling, and that is the signaling that tells your muscles to contract. So your muscles can get the signal to relax, but they can't get the signal to contract, leaving you paralyzed. Tetanus is the opposite. Tetanus goes in and blocks a neurotransmitter called GABA, which is the signal that tells you to relax. So with tetanus, your body receives a contract signal, or your muscles receive a contract signal from your brain, but they don't receive a relax. So with tetanus, you're spasming, your muscles spasm, and with botulism, you're paralyzed. So with botulism, the exotoxin is called botulism toxin, or Botox. For tetanus, the toxin is called tetanospasmin. It causes you to spasm. So really, they produce in exactly clinically opposite ways, just because of which neurotoxins, which chemical signals are being blocked by the exotoxin. Okay, so all of these, all of this being said, how do you treat an exotoxin-based disease? Well, the first thing that you want to do is stop the infection from getting bigger. So you give antibiotics. You also want to trigger the immune system. So if you've got a vaccine against these organisms, you give them the vaccine so that your body will start creating antibodies to grab onto and neutralize any exotoxins that are there. You need to stabilize your patient, make sure that, any symptoms that are there, they're treatable. You treat them. And then for many of these, we also have passive immunity. We have antibodies that we can give to the patient that will grab on and neutralize any exotoxin that are currently floating around while we're waiting for your body to start producing its own antibodies. So the type of vaccines that are specifically targeting exotoxins are called toxoid vaccines. You don't want to actually vaccinate somebody with exotoxin. You want to insert, I'm going to give you a tetanus vaccine. Here's some tetanus exotoxin. Hopefully it doesn't do damage. So we don't want to do that. So basically we have attenuated attenuated exotoxins, essentially. They're called toxoids. They're kind of like dummy exotoxins, and that's what's in a toxoid vaccine. So you give a exotoxin-like substance that your body then creates antitoxin antibodies against. So you've got, you get your tetanus shot, you get antibodies against the tetanus exotoxin produced, so that if you do get Clostridium tetani endospores in your body and they start producing the tetanospasmin exotoxin, you will have antibodies there ready to grab the exotoxin before it gets to your brain and starts causing the tetanus symptoms. Okay, so sorry this summary video was a little bit long, but there are lots of diseases to go over today, and I wanted to make sure that you got a nice kind of overview, wrap it all up, make sense of all the different pieces of material in this lesson. As usual, if you have any questions, please post them to the Muddiest Points discussion board, and myself and your classmates will try to help answer them. Good luck.